It's almost like I know that. This is like actually my screen. I just started seeing like how we click on it and no, they said so I'm like, I had to take that too. So I'm like, like racing the case. No, she's like, that's funny, but it's not. And you just take the management. The pharmacist, you know, next to the pharmacist has to correct herself. Like Joan, right? Like Joan. Like you're not saying. My description, and I get it, but I think my so like so much happening. I think that she wants to read still nerve-wracking. I just want to change it right. That's what he said, 4 packs but not 3, so I don't have to worry about it.
Does everybody have the 2 form? That's you?
Yeah, pass
Pass them.
Huh? What? Yes. I mean, she does. I said that. Excuse me. When they curse, they use the but just like they don't want it. But it's not, maybe I don't work with.
Okay, lecture 37. So Tuesdays, we have time to come in and chat. Review section at 5 for those who want it. If you need other times, this is the week that you can get them. I have not as many meetings this week. So if you need another time, let me know. There might be some opportunities on Wednesday and Thursday that I could meet with you if you need me to. Dam 2, Dam 3 is on May 2nd. Those are your lectures. I think everything's posted now. We've talked about multiple choice, matching, and sequence as a way of testing. I should be able, what are we doing this Friday? I should be able to get here by 11:30 to get started, and I'll stay as long as we need to. So, any questions on any of that? Everybody got the handouts that they need? Yes. As soon as I can get them done. Other questions? Okay, so we're going to talk about muscle function today, talk about special cases on Wednesday. We got an exam on Friday. So I'll again be using the same color scheme I had before. Again, you don't have to match these colors. Just know that things are different. We will make sure that all of the final metabolism schemes are posted with those colors so you can recopy, copy, whatever you need to. So using metabolism, we ended last time with where does acetate go? So in our diagram here, as a review, where does acetate end up? What are the potential fates, potential destinations for acetate following our diagram here? Yes. Yes. And what happens to it there? Acetate becomes a building block for our triglyceride. So the adipose tissue will make triglycerides. We're saving those energy units for a later time. You have one up here?
No.
Anybody else? Where else can acetate go? The liver. What does it do in the liver? What do we see in that diagram? Goes to oxidation. What does oxidation do? Generates the energy we need to run the liver. So we're going to the adipose tissue. Going to run the liver. If you probably want it to be complete in that, it could be part of the muscle going directly to oxidation, but we did not put that up there. Where does propionate go? What does the horse do with propionate? Stored from the cecum, where does it go? To liver. What does the horse do with that? What happens to it in the liver? Yes. Used to make glucose. So you're going to get very little propionate going by the liver. It's going to come from the gut capillary beds through the portal vein, open up again into the liver capillary beds. The liver is going to be very good at scavenging that propionate. It's going to turn all of it, well, most of it for this class, into glucose. So if you're going to follow propionate through the body, you have to follow glucose. So where does glucose go from? Well, in the liver first. What does the liver do with glucose? They can export it, or it can store it for later. How do we store it for later? Three things we do with glucose: build, burn, or store it. How do we store glucose in the liver? Right up there. What are we doing with glucose? Yes. Making glycogen. We're carving up. Saving that glucose for when we might need it later. So you've got the anabolic process in the sense that you're building molecules bigger. So glucose is going to go through genesis to form glycogen, and that'll respond to the hormones when we need glucose in the blood. Those hormones go there and tell the liver either make more or free what you've got. And then that glucose goes out into the bloodstream, goes to the heart, the lungs, the heart, and then can go out through the whole body. So from there, where does propionate end up? Anywhere glucose ends up. So where does glucose end up? Here to help make triglycerides when we have lots of extra. Here to either be stored or burnt, so build or burn. Or we're using it to run the nervous tissue, the brain. Brain has a preference for glucose as its fuel. Everybody following all that? One more potential thing, we think about the glucose, it could go through and be used as a carbon donor to make amino acids. So if that glucose, the propionate becomes glucose, glucose becomes amino acids, we got a whole nother range of things we can do with amino acids. Anywhere we build protein in the body, that propionate could be part of that. Butyrate. What do we say about butyrate? What is butyrate used for primarily? At least we're going to say in this class. Yes. Probably isn't going to get all the way to the liver. Where is it going to be oxidized? Where it's going to be used? Butyrate, 4 carbons. We want to oxidize it. What do we have to do? Break it in half. 2 carbon units go into oxidation, and we think about butyrate as helping run the gut. So it's not perfect with all this, but for what we're dealing with in this class, those discussions are good enough. If you go to vet school, they'll tell you a couple different things. We're trying to simplify things. So butyrate is going to be split in half and used to run that hindgut, provide energy for that hindgut to do what it needs to do. Is everybody starting to get a feel for how this works? If all else fails, follow the arrows. So amino acids, they're absorbed from the small intestines. We're going over here as part of the pool. Where do they go from there? They might make protein going up the synthesis side or exported into the body, and we can go anywhere we need to. We just talked about this, the muscles doing protein synthesis. Think about the myofibrils. As we build, as we train, those myofibrils get bigger and bigger. We get more of them. We're going to need amino acids to do that. So how is glucose made from this diagram? So we're not getting a lot of glucose directly to the liver. A lot of the glucose that's absorbed here is going to be turned into lactate. We're not going to absorb any glucose down here in the cecum. So if she's got glucose in her body, she's going to need to make it. We make it right here. So what goes into that to make glucose? We said 5 things. Propionate's the big one. Lactate, we talked about already. Anybody want to guess the other 3? Pyruvate, which comes from where? Anybody know? Glycolysis. Glycolysis, we break glucose down to 3 carbon units per pyruvate. We run it backwards. Gluconeogenesis. We get those glucoses back. We can steal the glycerol from fat prep. So fat prep going down here. Glycerol with gluconeogenesis. And then we have our non-essential amino acid can be part of that process. So those 5 3 carbon units can all be used to make glucose. The big one we'll talk about is that propionate coming in from the hindgut fermentation. So I think I put those in your study questions. We'll keep going. So in support of motion, we need to have those muscles working as best we can. So we're going to move over to the other diagram that I handed out, and everybody got a copy of this. So your diagram in your book here has a lot of the same information, but I didn't like the way it was presented. There's a lot of, well, that arrow's not quite where it needs to be. There weren't things separated as they should be, but it has a lot of the same information. After we go through the way I'm going to present it, you should be able to follow what's going on in this diagram. So we're shooting for this at the end of it all. So let's build it up one step at a time. So if we're looking at our diagram here, we're focusing on the muscle. What's going to happen there to support motion, to keep that muscle contracting, to keep that animal moving forward? So we have the gut, we have the portal vein. We know everything that's absorbed up here ends up in the liver. The liver is going to process those nutrients. Some are going to be converted, some are not. We're going to need the lungs to support those muscles. We have fat up here, adipose tissue. That's going to support the muscle as well. And then everything that's happening in that muscle to help move that animal forward. So let's talk about the digestive tract first as we move through this diagram. So we've got the parts there. What we have leaving the liver, either modified or not, is what we have to work with to make that muscle move forward. So we'll start with triglycerides. So the fat that's formed, you can say that represents a large category of fatty acids, triglycerides, non-esterified fatty acids, or all that stuff there, but we're just looking at fat. We'll use TG to represent that. We know amino acids are going to be digested in the small intestine, absorbed, brought to the liver. We know the liver is going to change some of those. The liver is going to release some of those by breaking down protein. We know the liver is going to take some of those and make protein. But if we're going to use amino acids around the body and other parts of the body, we need single amino acids. We can't absorb the whole protein. So we need single amino acids being released from the liver. So triglycerides, amino acids, we have acetate. Part of butyrate already. Taking all the propionate, turn it into glucose. The only BFA left is our 2 carbon acetate. The last thing that we're going to have coming out as a building block to move that muscle, move that horse forward, run the muscle, is glucose. We know we're going to have some absorption of glucose, not a lot. We know we're going to make glucose in the liver, that can be released. We know we have glucose stored in the liver, that can also be released. So somehow originating from our liver, and gluconeogenesis is going to play a part in having that glucose released. So what about the lungs in support of that motion? So we've talked about the lungs already. We know that oxygen is coming in from the lungs. Get that out into the bloodstream. So this is kind of an implied bloodstream, heart pumping around the body. We have oxygen getting into the blood, being carried by the red blood cells, and we're going to transport that oxygen, among other places. We're going to take it to the muscle. So we need that oxygen, especially for long-term activity, keeping those muscles running, keeping them generating ATP, keep them generating energy. We're going to need that oxygen. Most of that oxygen is going to go in support of oxidation. The last step in that cellular metabolism where we generate the most ATP. Oxygen is pretty important to the muscle, to the point where we have the ability to store oxygen in advance in the muscle. Has anybody heard of this before? We're dealing with a molecule called myoglobin. So myoglobin, very similar to hemoglobin, but instead of having 4 parts and can carry 4 oxygens like hemoglobin, we just got one heme here where we can get our hemoglobin or oxygen attached to, and those proteins are scattered throughout the muscle cell. Myoglobin, as a rule, is stickier to oxygen than hemoglobin is. So if you put them two together, the oxygen is going to tend to migrate over to the hemoglobin. Those hemoglobins are going to sit in that muscle and hold that oxygen for when we really need it. So you think about that horse coming out of the starting gate. You can't breathe in that oxygen fast enough. So having that there in advance to help generate ATP, go all the way through with glucose glycolysis, transition, oxidation. That oxygen is already there, all ready to go. So comparing the two, we've got a tetramer versus a monomer. We've got something that's roughly the same shape, the same sort of tertiary structure. We've got the same types of coils. If you dig into it, we know that myoglobin really wants that oxygen and is going to hold on to it strongly. We have those of you that heard about our little incident over in Auto. Everybody heard about that, right? So you've got hemoglobin is gonna, carbon monoxide is gonna stick right to that hemoglobin. That hemoglobin, it doesn't really want to separate from the carbon monoxide. That causes problems, and now you don't have sticky spots for the oxygen. So you have to go to the hospital. You're usually on pure O2 to help blow that carbon monoxide out. I didn't know they did hyperbaric. I started reading about all this stuff because I had an argument with my sweetie as to what exactly happens. I said, well, let's look it up and find out. So I thought carbon monoxide irreversibly bound to hemoglobin. No, it's just very sticky. So I was wrong, as always. So you have to get a lot of oxygen in there to displace and breathe out that carbon monoxide. And you usually need supplemental oxygen and hyperbaric chambers for like hours to bring all that out to get your oxygen carrying capacity back to where it needs to be. Because what the kids that ended up in the hospital, what is your pulse ox? It's somewhere around 90, 95, normally percent saturated. Those kids were down around 70. So they had to get them on supplemental oxygen. So we definitely need that there. We got supplies oxygen to the muscles only, helpful in the starving time of oxygen. So as that oxygen in those tissues drops down, myoglobin is going to give up that oxygen to help get through that oxygen debt until the lungs get caught up or fatigued, one or the other. And we'll talk about that on Wednesday. So we've got oxygen here. We've got some stored for later. Oxygen is going to be part of the oxidation phase. So you've got all these other substrates coming in, hopefully getting to oxidation to produce that maximum amount of energy per molecule going in. So cellular metabolism, it's all there. Coming out of oxidation, we're going to have CO2, carbon dioxide. We're also going to have carbon dioxide coming out of that transition phase. So we've got 3 carbon unit coming down. We pull one off during the transition phase. Don't generally generate energy. Then we've got 2 more being generated in oxidation. So oxygen goes in, carbon dioxide comes out from 2 other places. That carbon dioxide is going to flow down its concentration gradient, go into the blood where it's going to be moved in a number of different ways. And then we're going to dump that carbon dioxide in the lung. Everybody with me so far? Am I going the right speed? Too fast? Too slow? We have to remember, too, that as carbon dioxide builds up, it has a positive effect on the red blood cells dumping oxygen in that local area. As carbon dioxide builds up, we reduce blood pH. Remember the Henderson-Hasselbalch equation? That's going to make the hemoglobin less sticky. That less sticky hemoglobin is going to dump more of that oxygen right next to that muscle, right where it needs it the most. Any questions on the lung part of things in support of motion? Adipose tissue. I dropped out your amino acid here. We're going to focus on triglyceride, acetate, and glucose as going to the adipose tissue. So you should have those 4 in a row. We're just going to draw arrows. This should already be on your page, right? So we're going to just draw arrows from there. So triglyceride and anything related to that's just going to go to the, if we don't need it right away, it's going to go to the adipose tissue, the fat tissue, and be stored for later use. Whether that's tomorrow morning or 6 months from now. We think about a blue whale. Blue whales eat for 6 months and then lactate for 6 months. So they spend 6 months basically putting several elephants worth of fat away. Then they calve, or whatever it's called when you have a baby whale, and they spend the next 6 months transferring that fat back to that calf or that baby whale. So we can store fat that's pre-made that comes in from the diet. We can also make new fat. We can take the building blocks of acetate and glucose, and we can start absorb them into the fat tissue, and they become building blocks for the triglycerides. So we synthesized fat, our triglycerides. We have a new pool in there. We take our glucose, we split it into 2 glycerols, and then we start building fatty acids off those glycerols. So acetate, 2 carbon units. We keep building, adding to the chain. We add up even numbers of acetates, even numbers of carbons, and we'll go up to maybe 36 carbons in the chain. Everything from 6 carbons to 36 can be attached to that glycerol to form a triglyceride. That triglyceride, once it's formed, can go into fat storage. So I've got extra energy. I don't need the glucose. I don't need the acetate for anything. Let's store it for something that we can use later. Triglyceride is the best way to store energy on a volume basis. We talked about triglycerides being hydrophobic. You get a bunch of triglycerides together, they are going to group together and exclude water. If you have glycogen, that's going to be hydrophilic. It's going to bring water to it. So when you're thinking about the space it takes up per ATP or per amount of energy, triglycerides takes up a lot less space than if I converted all that energy to glycogen. If you took all this here and converted it to glycogen, I'd probably be out to here. So it's a good way from a volume standpoint to save that energy for later. We know that triglycerides, and this is a better representation than the last diagram, are constantly synthesized and degraded. A tub full of frogs. Things are going into the triglyceride pool, things are coming out. Everything's bouncing all over the place, wiggling all over the place. And when we break it down, we're going to go back to the BFAs and glycerol. We can turn around and move that back to triglycerides. So we're going to continue to evolve those triglycerides, change them up as they go through. In support of the muscle, once that triglyceride is made, and we're going through, say, winter, we need a little bit of extra energy to stay warm, we can pull that triglyceride out, put it in the blood, we ship it back through, it's going to rotate through the liver, and then come back around the other side and be useful for us when it comes to running the muscle. Everybody got adipose tissue. So let's get to the muscle and what it does. So this is what we've got so far. So we've got basic cellular metabolism. We've got the oxygen coming in, carbon dioxide coming out. What we're adding up here is the ability to store energy in the muscle for later use. You can store the fat right next to it, intermuscular fat, but we're actually getting to, with glycogen, we're taking that glucose, we're going to store it as glycogen, so this is inside the cell, ready to go. You've got oxygen inside the cell, ready to go. You've got glycogen inside the cell, ready to go. So if we need that quick burst of energy, we shouldn't have any delivery issues. Amazon has delivered everything. It's already at the house. So glycogen there, that glycogen, if I did this right, glucose comes in. It can either be burned, GTO, CO2 and water, or moved up here to be stored for later. This is carving up, getting ready for that big game the next day. You don't want to carve up too far in advance, or all that glucose is going to end up here as triglycerides. So we think about the spaghetti dinner the night before. What do you guys do to carb up your horses? Extra grain? Sure, somebody does something somewhere. So we've got glucose coming in. That could be our fuel for the cell. Either immediately, GTO, or we save it for the next day, next hour by storing glycogen. If we don't have enough oxygen, we can run glycolysis, which generates some ATP. And we talked about fast twitch muscles using this more extensively. If we don't have enough oxygen, we can't get down to oxidation. So we get a buildup of pyruvate at the end of glycolysis. Pyruvate is not something you want sitting around inside the cell. It's going to mess up the cell pH, start ripping things apart. So pyruvate cannot leave the cell, which has its benefits. So in order to manage that pyruvate, the body, the muscle cell changes it to lactate, which is exportable. Pyruvate can't leave, lactate can. We export that. That's going to go back to the liver and gluconeogenesis to get rid of that lactic acid, that lactate molecule. So what do we call that? Gluconeogenesis, glucose, glycolysis, lactate, gluconeogenesis. What do we call that? But remember. Or recycle. So we're net taking energy from the liver. We're transporting it here. Then we send that lactate back to get 2 more ATP. And we keep going around and around, moving our energy that we've captured from the liver to the muscle. Lactate can also go directly to oxidation in the muscle cell. So it's already the right number of carbons. It can feed right in and generate ATP directly going into our muscle cell. We can also run our muscle on triglycerides if we have enough oxygen. So you talk about those fast twitch muscles, they're going to rely more on fat. The slow twitch muscles are going to rely more on fat. Then the fast twitch muscle. Slow twitch muscles are good at getting that oxygen in, keeping that oxidation process going. So triglycerides are going to come into fat prep. Glycerol is going to go over to glycolysis or catch the end of it. So fat, GTO are fatty acids, reverse process of what we had up here. We're chopping off the acetates again. We chop them off and we feed them directly into our oxidation down low. So fatty acids go FO, the glycerol goes FGTO through, as we talked about before. So the last thing we getting from the liver being put into the blood, supporting that motion, we need to get amino acids in here so we can synthesize the proteins that we need to keep things moving, to keep things running. We need the structure. We can think of it as a truck made out of amino acids. And then we need the fuel, the gasoline. We can either run on fats or glucose. As a general rule, the muscle's not gonna, it can, but it probably is not gonna run on the protein. We're generally not gonna burn amino acids in the muscle to generate energy. We're gonna rely on the fat, we're gonna rely on the glucose. So this is gonna be very much downgraded, downregulated in our muscle. So we're going to use those amino acids preferentially for synthesis, creating the enzymes that we need, creating the contractile proteins that we need. We train that animal, we're going to need more amino acids to build more myofibrils, stronger, faster as we move through. You will have a lot of synthesis and degradation. It's kind of weird that there's so much energy involved in making proteins that it's weird that it turns over at all, but it is a constant process. So if we don't continue to train, we don't continue to use that muscle will wither away. Degradation will outstrip synthesis, and that muscle will contract from a protein base. Those amino acids, as we break down that muscle, can be released, put it back into the blood, and then they're going to go back to the liver and be part of that amino acid pool. So making new proteins, being shipped back out to go around the body, to be part of the transfer of amino acid or amino groups. One amino acid can become another as part of amino acid synthesis in the liver. So we're supporting our muscle. We're running our muscle mostly on triglycerides and glucose, depending on what we're trying to achieve. Aerobic, we're going to need a lot of oxygen to burn up those triglycerides. Down here, we can use glucose either anaerobically and use that in the Cori cycle to generate ATP, or we can take that glucose and burn it completely to CO2 and water. So fats, glucose, all the way to CO2 and water when oxygen is present. When oxygen is lacking or not present, anaerobic, we're going to basically live off of glycolysis, which doesn't require oxygen. We're not going to be able to do anything with the fat. It's going to sit there. Hopefully it doesn't build up and cause things like ketosis. So is everybody comfortable with the muscle? Is this too much, too little? So reminding you the Cori cycle, we pull that out. So gluconeogenesis with the lactate, that goes to glucose released into the blood. We go to the muscle. We generate ATP through glycolysis. We don't have any oxygen to burn all the way down. We release that lactate into the blood. That goes back to the liver and is regenerated into glucose. So we have a net transfer of 2 ATP per glucose to the muscle cell. So what do you think of that? Took me a long time to put this together, but I think I'm happy with the result. If nothing else, yes, sir. What I will probably do is ask you a question like I've asked already. Where does acetate go? Where do amino acids go? And for those of you that weren't here on Friday, I generally provide these diagrams. So you have the diagrams, you just got to be able to read them. And you should be able to answer the question. The nice thing that came with this here is now you have, and what I wanted when I put this together is you have continuous metabolism. We have the gut going to the muscle, going back to the gut, the liver, going around. So yeah, it's kind of odd that we had that, but I wanted to show, particularly like the Cori cycle, this continually runs around. So that's why I like this diagram better than the other one. Better than that one. You should be able to look at this from your equine science, and you can see they have all the VFAs here. VFAs, this to me, their placement is confusing. I didn't like the way that was placed. You got the Cori cycle here, no oxygen, lactate goes back to the liver cell, back to the blood, coming through. You've got the oxidation over here. You've got glucose. Glycolysis is going up. Got the transition when oxygen is present. The oxidation, carbon dioxide comes out. You've got different things being come through. So I just thought what I've generated, and it took me a while to get it done, was a little bit cleaner. So let's see if we understand what we've got. So here's our diagram completed. We'll get this posted if you want to print out a copy in color. I think last week's is part of last week's slides. This will be part of this week's slides, but I'll do each of these slides individually. So we curled it around. So how is adipose filled with triglycerides? What are the processes? So these are the questions I'm going to ask. So they're preformed, we dump them there. We can make them and dump them there. We can turn them over, mix them up, rearrange them, and dump them there. So making new, storing old, or pre-made. So how do the necessaries get to the muscle? Can everybody follow through how glucose gets to the muscle? How fat gets to the muscle? How protein is established, or amino acids get to the muscle, or oxygen? So glucose, how are we getting that to the muscle? Glucose regulated by the liver, put in the blood, transferred to the muscle cell. Triglycerides. How can we get them there? You've got the pre-made ones coming in on the diet, can go directly, especially under the influence of insulin, go directly to muscle cells. We can make some here in the fat, that's got to go through our circulatory system, become part of the triglyceride pool, that can come in and be used as well. Amino acids. How do we get our muscle protein? Amino acids from a variety of sources in the liver go to that muscle cell and are used to synthesize protein. Oxygen, we talked about the lungs. Providing that oxygen disappears, becomes CO2 and broken out. What are your thoughts on this? Like it? Don't like it? Did I go too fast, too slow? Okay, so we'll talk on Wednesday, special cases. Slow twitch muscles, fast twitch muscles, oxygen, anaerobic metabolism, aerobic metabolism. Thank you. Everybody sign the attendance. Yes.
If you're gonna put any grade into right space, just so we can see what our like full grade is.
Maybe if I get time, but that's not my strength. So, you're looking at, grade attendance is not required in this class, but I'm using it to get a feel for who's learning and who's not by showing up. The rest is 4 grades, and I hope to get, if nothing else, when I get the next exam posted, I'll give you a grade calculator, and then you can just put your grades in there and then project what you want for your final grades, and you should be able to go through. So,
Thank you. I
yes.
You said the sheets that we just did, you would put them in the exam, like we can use them as a cheat sheet, or did you mean like we had to fill it out?
No, I will give you this printout, that printout.
Well, can I just use the ones that we made in class because it's all colors?
We could talk about it next time if you prefer your own drawings.
Yeah, because I highlighted it.
Okay.
I didn't-
Okay, we can talk about that. I don't see where it'd be a problem. I just want to make it fair across all students.
Yeah.
So I'll have to figure out how we do that. Do I allow you one full sheet of paper and you can write anything on it you want? Or we just have metabolism on front and back? I can give you the blank ones and then you can fill it in the way you want. So I give you a blank front and back, you fill in the way you want, or I can give a color one pre-printed. So I'll have to think about that, but we can work on that.
Yeah, because the color coding helps personally me understand like what is what.
Okay. Yeah, we'll work on that. Okay.
Thank you.
You're welcome.